welcome to Med Madness. Today, we're diving into the world of antibodies. But first, let's test your knowledge with a USMLE Step 1 style question. During a research internship, you are assigned to work on an assay that detects the presence of a specific protein marker in serum samples using antibodies. Your mentor asks you to choose between using monoclonal or polyclonal antibodies for the assay. Considering the need for high specificity and consistency across multiple tests, which type of antibody would be more appropriate and why? A. Monoclonal antibodies because they bind to multiple epitopes on the antigen. B. Monoclonal antibodies because they bind to a single epitope on the antigen. C. Polyclonal antibodies because they are derived from multiple B-cell populations. D. Polyclonal antibodies because they bind to a single epitope on the antigen. Think about this scenario and we'll reveal the answer shortly. Let's break down the differences between monoclonal and polyclonal antibodies to help you answer this question. Antibodies are proteins produced by B cells that recognize and bind to specific antigens like bacteria or viruses, marking them for destruction by other immune cells. Monoclonal antibodies are derived from a single B-cell clone, which means they are identical and recognize only one epitope on an antigen. This makes them incredibly specific and consistent, ideal for diagnostic tests where detecting a specific antigen without cross-reactivity is crucial. Polyclonal antibodies, on the other hand, are sourced from multiple B-cell clones. This means each preparation contains a mixture of antibodies that can recognize different epitopes on the same antigen. They provide a broad response and are particularly useful when the detection of all possible targets is necessary, such as in certain infections or complex biological samples. Given that our scenario requires high specificity and consistency for the assay, the correct answer is B. Monoclonal antibodies because they bind to a single epitope on the antigen. Monoclonal antibodies will ensure that our assay consistently detects the specific protein marker without interference from other similar proteins. Now let's think broader. How might the choice between monoclonal and polyclonal antibodies affect the outcome of a therapeutic intervention? For example, consider a disease where the pathogen can mutate quickly. Which antibody type might be preferable in this scenario, and why? Drop your answers and thoughts in the comments below. Please like, share, subscribe, and visit medmadness.co to buy the full immunology course.